Hi everyone, today we'll be covering the IGCSE Human Biology 2022 May paper 2, but this is the R version, so 2R version. So this paper is worth 90 marks and you'll be tested on the entire syllabus. Okay, let's start off with the first question. A student investigates whether there is more carbon dioxide in expired air than in inspired air. This is the apparatus the student uses. So expired air is air that we breathe out, inspired air is air that we breathe in. So to test for the presence of carbon dioxide, we would use lime water, and lime water turns milky in the presence of carbon dioxide. So this indicator here would be lime water. Okay. So let's go on to part A. Draw a diagram to show how the apparatus should be put together for the investigation. So I'll draw it out first, and then I'll go over how the apparatus works. So we're given 2 conical flasks. And then we're also given a bungs with glass tubing. So there's a short end and the long end. The long end is going to go inside of the indicator, and we said the indicator was lime water, right? So this is lime water. Then we have the bung on both of them. We have the short end of the tube go here, and then we have the long tube that would go into the indicator. We also have this glass T piece here. So that's the part where the participant would put their mouth. So we would have this T tube there. And then again, the short end of the tube, and then the long end going like this. So in a different color, I'm going to show what actually is supposed to happen here. So this part is the mouthpiece. That's where they would put, the student would put their mouth onto the glass piece. And what would happen is air would come in from this part. So this would contain inspired air. And then the air would come up this tube and go inside the person's mouth. And then when they breathe out, it would leave this tube and enter this conical flask. So this conical flask will contain expired air. And then it would react with the lime water here. So because expired air contains a lot more carbon dioxide, this should actually turn milky. And this would be colorless. And then the air go back out like this. So the green annotations are just the stuff that I've added. You don't include the green parts in the exam. Okay, part B, name an indicator that can be used for this investigation. This would be lime water. And then part C, describe how the apparatus would be used to determine whether there is more carbon dioxide in expired air than in inspired air. So we've gone through this part earlier. So the participant would breathe in and out through the mouthpiece. For maybe about a minute, Then compare the contents of the flasks. The exhaled, or the conical flask, containing exhaled air would turn cloudy. You can say cloudy or you can say milky, which indicates that there is more carbon dioxide. Okay, let's go through the marks. So the first mark is breathe through the mouthpiece. That's one mark. In and out, that's a second mark for a maximum of 1 minute. That's a third mark. Then you would compare the contents of the flask, that's the fourth mark. And then the one containing exhaled air would turn cloudy. That is the which indicates that there's more carbon dioxide, so that's the fifth mark. Okay, state one safety precaution. So because you'll be using perhaps different participants, it's best to sterilize the mouthpiece because that would be pretty unhygienic. So the mouthpiece should be sterilized. All right, state one variable that should be controlled. So if we go back to this experimental setup, one thing that can be controlled is the volume of lime water that you've added to the conical flask. So the volume of the lime water in the conical flask. Okay, question 1 is done. Question 2. Okay, a student investigates the effect of a competitive inhibitor on the action of enzyme phosphatase. So this is the reaction catalyzed by the enzyme, so this would be phosphatase. So that's the enzyme that does this reaction. Phenolphthalein turns pink when an alkali is added. This color can be measured using a colorimeter. The more intense the color, the higher the reading of the colorimeter. This is a student's method. Prepare 6 different concentrations of phenolphthalein phosphate solution. Place 5 cm³ of each solution into a different test tube. Add 1 cm³ of the competitive inhibitor and 1 cm³ of the enzyme. Wait 30 minutes, then add 1 cm³ of the alkali to each tube. So the alkali is responsible for turning it pink. So it turns pink when in the presence of phenolphthalein, which is the product here. So this plus the alkali makes it pink. Then measure the intensity of the pink color using a colorimeter. Okay, so part A1, give a suitable control for this investigation. So the purpose of the control is to make sure that any changes is due to the independent variable that's being tested here. So essentially what you're doing is you're adding a competitive inhibitor to the phosphatase. So what you would want to do is have a test tube that has the inhibitor, that has the substrate, but doesn't have the enzyme. So have a test tube containing the inhibitor and the substrate, but not the enzyme. So if you were to have a test tube without with the inhibitor and with the substrate, but without the enzyme, you would expect it to remain colorless. If it just so happens to turn pink, then you know that the reason why it turns pink is not because of the enzyme. So this is just to rule that out. Okay, state 3 variables that should be kept in this investigation. So let's scroll back up so that we can see what we're dealing with here. So whenever we're dealing with enzymes, we want to make sure that number 1, the temperature, and number 2, the pH are controlled unless the temperature and the pH just so happens to be the independent variable. So that's one thing, you want to make sure the temperature and the pH is controlled. The reason why is temperature affects the rate of enzymatic activity. The greater the temperature, the more faster the enzyme works, and then if the temperature is too high, then the enzyme gets denatured. Whereas pH, if it deviates from its optimum pH, then the enzyme would be denatured. So those are the 2 things. So let's write this down here. So temperature is one thing you want to keep constant when it comes to enzymes. The other one is pH. Now, for other examples, we can go through the method itself. So do you see how in this step, we're adding 1 cm cube of the competitive inhibitor and 1 cm cube of the enzyme phosphatase? Not only do we want to keep the volume the same, but we want to keep the concentration the same as well. So that could actually be 2 separate marks for that. So you can say concentration of the inhibitor. You can also say concentration of the enzyme. That's another point you can also say. All right, part B, the table shows the students' results. Okay, so we have the concentration and then the intensity of the pink color. Okay, because we're changing the concentration of the phenolphthalein phosphate solution, that's going to be our x-axis. So concentration of phenol, oh, that's pH. Phosphate solution. This is in arbitrary units. So arbitrary units. So the largest number here we see is 0.8. Let's try and see if we can fit 0.8 in this axis. So we can have here 0.0. This is 0.1, 0.2, 0.3, 0.4, 0.5, 0.6, 0.7, fits well, 0.8. And then we have the intensity of pink color in arbitrary units. So this is going to be quite tricky to draw. There's not enough space. So I'm just going to draw outside, but don't do this. Intensity of pink color. So this is in arbitrary units. The largest number that we have here is 0.91. So let's try and fit that on the graph here. So we can have it go up in 0.2. So we'll have 0.0, 0.2, 0.4, 0.6, 0.8, and 1.0. So long as it takes up more than half of the space provided, then you're good to go. So let's look at the first point. The first point is 0.5 and 0.2. Sorry, that's 0.05 and 0.2. So 0.05 is here and 0.2 is here. Next we have 0.1 and 0.3. 0.1, 0.3, that's right there. Next is 0.2, 0.47. 0.246 and 7. So this is 0.5 right in the middle. So it's going to be less than that. Okay, the next part is 0.4 and 0.65. So 0.4 and 0.2465. It should be somewhere in this middle here. Then we've got 0.6 and 0.79. So 0.6, 0.79 should be in between there. And then we have 0.8 and 0.91. Okay, so it says draw a curve of best fit. So I'm going to attempt to draw a curve here. Okay, there we go. All right, the next part, part 3, add a second curve to show the results to show the results that would be obtained if the competitive inhibitor had not been added. Okay. A competitive inhibitor is an inhibitor that competes for the active site of the enzyme. And so because of that, it has a very similar shape to the substrate itself. So if you don't have the competitive inhibitor, then that means the rate of reaction would occur a lot faster. And so this is what the curve would look like. Okay, let's move on to part C. Describe how the results of the investigation would be different if a non-competitive inhibitor had been used instead of a competitive inhibitor. So I've included a diagram to showcase the difference between the competitive and non-competitive inhibitors. So on the left we have competitive inhibitor, and you can see the competitive inhibitor is competing for the active site. So it'll bind to the active site, whereas here on the right, we have a non-competitive inhibitor and it binds to the allosteric site, so not the active site, and that would cause the active site to change shape. So now the substrate can no longer bind to the active site. Now, because of that, because the active site has changed shape, then that means the maximum activity cannot be reached. So I'm just going to draw this in a different color because you don't have to draw this for this question, but I'm just going to draw what it would look like. So it would look like, sorry, that's a very squiggly line. So it should look something like this. So the maximum you can see cannot be reached when you're using a non-competitive inhibitor compared to a competitive inhibitor. So I'm going to remove those annotations and write that down. Let's make this one smaller. The maximum enzyme activity cannot be reached even as the substrate concentration increases. Okay, let's move on to question 3. The diagram shows two types of blood vessels, A and B. Draw an arrow on the diagram to show the direction of blood flow in vessel B. So let's identify the blood vessels first. Vessel A is the artery, and blood vessel B is the vein. The reason why we know blood vessel B is a vein is because of this valve. So valves prevent the backflow of blood. Let me just erase that. Actually, before we continue, let me include a little diagram here to show you how a valve works. So here we have the valves of a vein. So we can see here, these are the valves, right? So as blood is pushed forward, it allows for blood to flow through, but then blood can't flow backwards because the valves would then close. So it's like a one-way door, essentially. So if we were to answer, let me just remove this. So if we were to answer this question, the blood flow is moving from here downwards. Okay, part 2, identify each type of blood vessel, give explanations for why there are differences in their structures. So vessel A, we said earlier, is an artery. So we can see based off of the walls, if you compare the walls of the artery to the walls of the vein, the walls of the artery are much thicker, and the reason why they are thicker is to be able to maintain high blood pressure as blood is being pumped from our heart. So vessel A is an artery as it has thick muscular walls. This is to maintain high blood pressure as blood gets pumped from the heart. Okay, blood vessel B is a vein. Vessel B is a vein as it contains valves. Whoops, contains valves to prevent backflow of blood. All right. Part 3, state the name of one other type of blood vessel, that is your capillaries. So the capillaries are responsible for nutrient exchange in your tissues. So you would say capillary. Okay, moving on to part B. Coronary heart disease kills many people each year. The graph shows the number of deaths per 100,000 of the whole population in a part of the world. So for part 1, it says describe the trends shown by the graph. And usually when we come across these types of questions, we would talk about how whether or not there's a relationship between the x-axis and the y-axis. So you would say, oh, as the year goes by, there is a decrease in deaths. And this is a trend that we see in both males and females. So let's write that down. We can say from 2005 to 2014, it's a decrease in deaths for both male and female. So it'll be 1 mark for this, 1 mark for decrease in deaths, and the last 3rd mark is for both male and female. Okay, let's move on to part 2. The part of the world for which the data was collected had a population of 60 million people in 2006. Calculate the number of men who died from coronary heart disease in 2006. Okay, let's go back up here to this graph. Okay, so according to the graph in 2006, there is 640 deaths per 100,000 of a population. So I'm going to write down a little ratio thing here. So deaths and people, or like population, right? So we'd have 640 deaths per 100,000 people. And it says here that the total population is 60 million. So I'm going to write 60 million. So what I do, because it's a ratio question, so what I do to this number, I have to do to this number. So in order to get from 100,000 to 60 million, what I could do is divide by 100,000 to get 1, and then times by 60 million. Ideally, you would write all of this down in the space provided, but I want it to be closer to the graph. Okay, so let's do 640. 640 divided by 100,000 times by 60 million. That should give me 384,000. So what I did on the right hand side, I have to do to this number as well. Okay, so I just checked the marking scheme, and the marking scheme is incorrect for this because it says 38,400 when the answer should be 384,000. Yep, so the marking scheme is incorrect for this. All right, let's go on to question 4. State 3 methods of birth control. Okay, so I'll list them first and then I'll briefly go over them one by one. First one is IUD, that stands for intrauterine device. Second one is contraceptive pill. The last one is a barrier method, so you can say things like condoms. Okay, let's briefly go over them one by one, starting with IUD. So here we have an example of an IUD. So IUD stands for intrauterine device, and we would insert that into the uterus. And so you've got hormonal versions, but you also have the copper version. So the copper version just prevents sperm from fertilizing the eggs. That's how your IUD works. Let's remove this one. And so this is an example of a hormonal method. This is the contraceptive pill. So we have contraceptive pills that would essentially prevent ovulation from occurring. You could also have an implant as well, which will slowly give off hormones like progesterone to prevent ovulation. So there are different ways to go about releasing these hormones. And then the last one is barrier method. So here are some examples of barrier methods. We have the male condom over here, and then we have, this is a diaphragm, so you would insert that into the vagina, and both of these prevent sperm from entering the uterus and therefore entering the oviduct, where fertilization would occur. Okay, let's move on to part B. The graph shows the changes in the width of the uterus lining in the concentration of estrogen in the blood during one menstrual cycle. And then part 1, it says determine the change in width of the uterus lining between day 6 and day 14. So day 6 is here, day 14 is here, and because we're looking at the width of the uterus, we need to look at the circles here, and we would need to look at the axes on the right-hand side because that says width of uterus lining in millimeters. So we can see here for day 6, this is about, so this is 0.5, this is 0.6, 0.7. This spot should be about, or this dot should be about 0.65. Now let's look at this one over here. So this is day 14, and this is 4.0. So it should be right in the middle of 4.5 and 4.0. So this should be 4.25. In the marking scheme, it says 4.35, but I believe it's 4.25 because it's just right in the middle of both of these two numbers, 4.0 and 4.5. So let's write that down. We have 4.25 minus 0.65. So let's write that down. Calculate that, so that should equal 3.6. So 3.6 millimeters. Okay. So part 2, it says explain the importance of this change in width. So I'm just going to include a diagram. I've included a diagram here for your reference, but you don't need to know the names of some of this stuff, but you do need to know fertilization. So fertilization is when the male nucleus and the female nucleus fuse together. So in this case, the nucleus from the sperm and the nucleus from the egg fuse together. And then you'd form a zygote, and then it would undergo mitosis to form an embryo. You don't need to know these names. An embryo, and then the embryo would then implant itself into the lining of the uterus. So it's very important for the uterus to be, or the lining of the uterus to be thick to allow for this implantation to happen. So let me just remove this. Oh, I think I removed this by accident. Oops. Okay. Explain the importance of this change of width. So the increases so that if fertilization occurs, then the embryo into lining. Okay, determine which day menstruation occurs. So menstruation occurs when you have the shedding of the lining. So in the marking scheme, it says that menstruation happens between day 0 and day 6. So this is because estrogen levels would fall, and progesterone, so both progesterone and estrogen levels would fall right before menstruation. So that's why it's day 0 and day 6. But I'll be honest, this graph has me a little stumped because I would have thought that menstruation happens somewhere here where there's a drop in the width of the uterus lining, but it says it's from day 0 to day 6, so that's just what we're going to write here. So from days 0 to 6. And then part 4, ovulation takes place on day 15. Describe how the two lines of the graph would be different. Describe how the two lines of the graph would be different if the ovum had been fertilized. So we need to describe what the lines would look like. So if the ovum had been fertilized, then that means that the uterus lining needs to be thick in order for implantation to occur. And the uterus lining would remain thick until the end of the pregnancy, right? So that means that the lines for a width of uterus lining, it would remain high, so probably remain somewhere here. And then your estrogen concentrations would also be high as well, or level off, because estrogen is important for, number one, ensuring the growth of the uterus lining, but also number 2, to prevent more ovulation from occurring. So that would also level off, but at a lower level. So let's write that down here. So endometrium, or the uterus lining, would level off or would plateau. All right, question 4 is done.